So in this slide, which is from page 87 in your textbook, we see a really interesting some data from the firing rate of neurons in the inferotemporal area. And whenever you record from neurons in the IT area, of the cortex, and present faces, this is in monkeys, right? You present them faces, even human faces, you get a very high firing rate here, as opposed to non-faces like flowers and buildings and things like that. Here's more data with comparing the firing rate, looking at these face selective cells, right? So these are recording from these cells again, right? You'll see they fire like crazy to faces, and then after we show them a bunch of faces, we show them some bodies, right? The whole body, not just the face, but the whole body, and firing goes way down. Fruits, firing goes down, although it goes a little higher, I guess, because they're hungry, maybe. Gadgets, objects, things. Hands, right? And then scrambled, right? Scrambled up, scrambled faces, scrambled up faces, and scrambled up images all mixed up together, right? You'll notice that uh, you only get the selective firing in the IT area to faces, right? Humans, we can look inside of a human brain using fMRI techniques, right? And the subtraction technique. If you remember that, that's where we do a baseline recording using MRI, and then we present a stimulus and record while the stimulus is being presented, and then subtract the difference for each brain area to see which brain areas are most active. Humans have a part of the cortex called the fusiform face area that responds absolutely best to faces. We have a parahippocampal place area that's right around our hippocampus that responds to spatial layouts. Whenever you're seeing objects laid out in space, like inside of a big room or whatever, this area lights up quite a bit. The EBA, the extrastriate body, responds best to pictures of full bodies and body parts like arms and legs. Okay, so it's really interesting that we have these modules in our brains that are specialized for detecting certain stimuli, right? So, for example, here's the parahippocampal place area, right? So this is our hippocampus here, and this is right next to it, right? The parahippocampal place area responds to spatial stimuli, so images where we're looking at objects laid out in space, right? So that area lights up for these types of images, but not so much for these types of images where these aren't really uh, objects represented relative to each other. They're just simple flat objects. The the extrastriate body area, EBA, responds best to pictures of full bodies or body parts. So it likes pictures of arms and the body, and even a stick representative of a body or a shadow representative of a body. But it doesn't respond to objects like hammers, a face, a mixed up stick, or a mixed up silhouette of a body. Here we have some data where we're looking at different types of activation from different types of stimuli, right? So basically, and these images here are kind of like your weather map, where the very yellow orange are going to be very intense, and the blue green are not so intense. Um, you can actually segregate by different categories which parts of the brain are more active, right? So, for example, for faces, right? If we look at faces, right, we see that the faces light up here in the fusiform area, right? Um, for chairs, which are just objects, right, lights up over here. And for houses, more in the parahippocampal striatal area here. Let's see. The the structures in the limbic system are very important in regulating memory. The medial temporal lobe, that's what MTL stands for, medial temporal lobe, which includes parts of the hippocampus, a large part of the hippocampus. And we know this from a patient named HM. HM was a person who had very severe temporal lobe epilepsy. So it was epilepsy that started in his temporal lobes and spread to his brain. And they could not treat it with anything, so what they decided to do was to remove his temporal lobes. Well, when they did that, they also dug in pretty deep and removed the hippocampus as well, which goes into the temporal lobes. And it turns out that HM was no longer able to learn new things. So he could remember most of what he had learned up until the point before surgery, but after surgery, he was completely unable to learn new things, to learn new people, new places. He couldn't learn them anymore because his hippocampus was destroyed. He recently passed away about maybe 5 years ago, and it was a big deal on the internet because his identity was finally revealed, and they actually took his brain and sliced it, and they're studying it right now to see exactly what parts of the hippocampus of his hippocampus were damaged. Um, if we look here, we have parts. This is a view under of the from looking at the brain from the bottom up. So if I were to take a brain and you were and flip it upside down, you would be looking at the bottom of it. So this would be where your eyeballs would be, right about here, and this would be your forehead here, and then the back of your head here, and the side of your head here, of course. And we have these areas: the perihippocampal cortex, the entorhinal cortex, the amygdala, and the hippocampus. These are all these areas that are in the medial temporal lobe and in the limbic system. Very important areas associated with memory. And we have in fact identified cells in the hippocampus that fire to specific images. For example, there was a study that was done where a patient was who was having their hippocampus recorded from they found a cell that responded to Halle Berry, to pictures, images of Halle Berry, which I think is kind of cool, right? There was another study that was done of a neuron that uh, in the in the hippocampus, where we asked people to recall things, right? So, uh, you know, recall something about New York, think about the Hollywood sign, think about the Simpsons, right? And then they laugh, right? And but as soon as they're told to think about the Simpsons, this particular cell fired like crazy, right? So it was like a Simpsons cell inside of our hippocampus, right? Um, we're not hardwired. Okay, that's one of the really important things you learn from this. All of these modules grow and develop with experience, right? So obviously there is a certain level of architecture that is involved in connecting all these areas together and creating them so that they are specialized somewhat. But what they actually respond to across the lifetime of an individual is determined not only by the way they're wired, but also by the experience, right? That a person has to train or to allow these areas to acquire their specialized skills, right? Um, so, for example, we can take normal people and train them. We can train up their FFA, their fusiform facial area, to recognize um, certain stimuli that they couldn't recognize before, right? So this right here is what a guy named Gautier created this little sort of pseudo face here called a greeble, right? And he had all these different kinds of greebles here. And what we first did was ask people to look at these and measured their MRI response in their fusiform face area. Now, before training, these things didn't really light up the facial recognition area, the FFA area, as you can see here in the first set of bars, right? Faces lit up the area quite well, of course, because it's the fusiform facial area, but the greeble faces, these fake faces here, did not. However, after people were exposed to these many of these faces and trained to recognize them, uh, they became faces, right? We started recognizing them as faces, and you can see after training, the greeble faces were recognized almost as much as human faces were, by in terms of the response of the neurons.